So good afternoon, dear Vice President Spiegel, dear colleagues, dear Timothée. As current department head of the Department of Information Technology and Electrical Engineering, it is my great pleasure to introduce our new colleague, Timothée Proix, who joined us last year in August as Assistant Professor of Computational Neuroscience. Timothée grew up scientifically in France, where he completed his master's degree at a Marseille University before pursuing his PhD degree at the Brain Dynamics Institute at the same university. His doctoral work focused on large-scale modeling of epileptic seizure dynamics. After his PhD, he moved to Brown University in the United States for a postdoc, before returning to Europe and spending several years at the University of Geneva, where he was awarded an SNSF Ambizione Fellowship, with which he was able to build his own research group. His research asks a question that sits at the heart of what makes us human. How does the brain turn sounds into words and words into sentences? To answer it, he combines theoretical modeling, machine learning, and direct recordings from individual neurons in human patients. The epilepsy threat is a common theme in his research. His earlier research produced advances in understanding how seizures propagate through the brain and in developing models that can forecast when a seizure is likely to occur. That work earned him the Alfred Hauptmann Award from the Swiss League Against Epilepsy, as well as the French Society of Neurology's Excellence in Neurology Award. Recognition from two different clinical communities demonstrating the real-world relevance of his science. With his arrival, our department strengthens its presence in theoretically grounded neuroscience and opens new connections toward machine learning, neuromorphic computing, and biomedicine. These are exactly the kinds of bridges that make an electrical engineering department more than the sum of its parts. We are genuinely delighted to have him here, and please join me in welcoming Timothée Proix.
Thanks a lot, Gabi, for the very nice introduction. It's an honor to be here today and to give this lecture in front of you. And today I would like to go through the questions that drive the research in the lab, which is at the Institute of Neuroinformatics at the Inaugural campus, as you can see here on the photo. And also I would like to tell you about what we're trying to understand about the brain and how it handles language and what we have found so far. So let me start with a small experiment where I will give you this sentence, which is, the retired neuron politely declined the linguist who had organized a fondue evening. And so this is a new sentence that you probably never heard before, and yet you were able to understand it as I was saying it, and you may even have pictured this sentence in your head. And it took you a very little time, probably less than a second. So this is the puzzle I want to start with today. How is this possible? How can your brain, which is a finite biological system, can make sense of this sentence and understand it, even though it never seen it before? And basically this puzzle has a name, which is the infinite use of finite means. And these names come from the linguist Wilhelm von Humboldt in the 1830s. And basically, which means that we have a finite number of words, of rules, and of different phonemes, but you can combine them to make infinite number of sentences. And to give you a concrete example of this, you can take the words I had in my sentence just before and reshuffle them to make new sentences. For instance, a retired linguist politely declined a neuron who had organized a fondue evening, or the retired neuron politely organized the linguist who had declined the fondue evening, or even worse, the retired fondue evening politely declined the linguist who had organized a neuron. Right? So from each of those sentences actually grammatically correct, and they mean something different, and they are very likely new. They probably never were said before, except maybe when I was preparing this talk. So from a small set of words and rules, you can really generate infinite space of meaning, and it's what we are trying to explain throughout our research. So I would like to approach this puzzle through three different questions. And to give you an example, if I take the sentence, the cat chases the dog, we go from sounds that we hear to a meaning, which is here represented by this image, the cat chasing the dog. And the first level that we want to look at is the level of the representations. We need to represent the different elements of the sentence in the brain with neural activity. For instance, using the different phonemes, K, A, and T for the cat, or the actual word cat. We also need to compose those different elements from lower level, let's say the K and T, to compose them into a bundle and make the word cat, or to take the cat, the dog, and chasing and put them together in a sentence that actually means something. And then all of these levels are actually interacting, because when you understand this sentence, you don't build up from one level to the next, but you actually do it on the fly. And there's this interaction not only from the bottom up, but also from the top down. And so in my lab, we try to study these questions through a combination of recording methods, where we record neural activity from the brain, but also from modeling. And these three different questions will be the three sections of the talk I will give you today. But before, I would like to give you a little bit of an introduction about neurobiology of language, and also how we can actually record this neural activity. So for decades, researchers have been trying to record the actual brain activity, in particular using something called functional MRI. And where using these methods, they can actually do very nice mapping of the brain activity corresponding to the different functions that are involved in language. So if you start from the brain and you hear, let's say, some sound corresponding to a sentence, you will first process the sound in you can see in green, the hemisphere gyrus, which is the dorsal part of the supratemporal gyrus. And then you will recognize the different phonemes, doing phonological analysis in the supratemporal gyrus. Then there will be, of course, this simplified scheme, but you'll do some semantic processing along something we call ventral stream, and then syntactic processing as well, and all the way to articulation when you actually speak. So here we have a very nice map that tells us where those functions are processed, but does not really tell us how it's done, And this is what we're trying to understand in the lab, how these computations are done. And for that, we actually need to record brain activity, which is a much higher temporal and spatial resolution. And we use, for that, a rare clinical opportunity, which is from patients with epilepsy. So those patients, they usually, some of these patients actually don't respond well to medication. And for those patients, sometimes it's a good idea to try to actually remove the brain area from which the seizures come from. And for that, they come to the hospital for 1 or 2 weeks, and then the neurosurgeon will implant some electrodes in the brain, and then try to figure out where those seizures are coming from in the brain. So this is an example of X-ray of such a patient, where you can see in black the different contacts of those electrodes, and it actually looks like this, right? You have this stick, and each of the white part here corresponds to an electrode contact that can record the local field potential of the neural activity. And you can see on the right, there is a MRI and structural MRI, and you can see this white bar that corresponds to the electrode recording the brain activity. And of course, when the patients come, we don't record a single electrode, but actually a couple of those electrodes. And this is what you see here in different colors, and each of the dots correspond to one of the recording contacts. So with this kind of data, it's very nice, because we can record in very high temporal and spatial resolution the brain activity. And to show you an example of this, I take an example from this patient. Here you see the X-ray, which had death electrodes that I just showed you before, as well as what we call electrocorticography, or surface electrodes that are on the cortex, but also invasive recordings. These are all of the white dots you see here on the surface. And also non-invasive EEG electrodes, which are placed on the skin. And this is actually a case reported in this book that is one of the first books I read during my PhD from Michael Sperling in 1993. And it's a very beautiful book, because when you open it inside, you have all of these time series showing you the recordings of the different electrodes in the brain. And at the arrow, you can see that there is a recording of intracranial activity, and you can see for that patient, there is a seizure starting at where the black arrow is. As you go through time, this brain activity is going to evolve and increase for that particular electrode. Then if you go even 30 seconds later, you can see now this is the contact, the blue contact here, intracranial, is on the surface of the cortex. So far, nothing is happening. But if you go a bit later, you can see that now you have activity growing in this electron. And at the same time, in this patient, they were also recording on the surface of the brain. And if you look, you don't see anything on this particular contact. And you can go even further, see the seizure actually propagating and evolving, propagating in different regions, and even becoming very large amplitude on the cortical surface. Yet, if you are not a trained neurologist, you won't see anything on the surface electron. And so at that point, I decided I don't want to do surface EEG, but intracranial EEG for my studies. And And so basically, what I showed you is epilepsy recording. So this is another example of such a recording of seizure, which is called a complex seizure, where you start from a particular brain area here, and then propagate throughout the brain in different other contacts. And this very prominent, very large amplitude type of activity. And this is actually some part of the work I did during my PhD and my first postdoc, where I tried to model with dynamical system the propagation of this activity throughout the brain. But when you look at physiological neural activity, normal activity, for instance, during speech processing, this is very different, right? You don't see any large amplitude peak. It's very hard to understand what's going on or to actually figure out what's going on. So for that, we can use tools, for instance, dynamical system type of tools that we use both in epilepsy and for normal neural activity, or other kind of fancy analysis. And so what we are interested in is indeed to look at speech and to record natural speech. So how do we do this? The patient is there in the hospital for 1 or 2 weeks. And then we record what they say. It could be either like natural conversation that this patient has with other people in the room, so hearing some speech or actually speaking, or it could be actually experiment that we propose them to do. And this is an example of data set collected over several years across several patients. So each of the colors correspond to the electrodes for a single patient. And because these patients are staying there for several days, we can actually record the brain activity and the conversation at different moments of the days. So this is an example of such a recording. You can see it's going across 9 days. And all of the colored bars correspond to actual neural features that shows some actual recording of the speech activity. So of course, there are many moments where the patient is not speaking or it is sleeping. So if I just put all of this together, you can see more nicely that we collect up to 50 hours of speech for that particular patient. And so we can then go into this data and try to figure out what is going on, can ask many questions to this data. And so here what I will show you today is some glimpse, some small sample of this long data set. One thing we can also do is to try to go even smaller scale, and instead of recording the local field potentials, we can record single neurons. And this is possible because we have other types of electrodes. So this is an example of microelectrode UTA array that was implanted in the brain of this patient. So it's a very small 4x4 square millimeter electrode array. And each of those pins here correspond to contacts that goes inside the cortical surface or the cortical sheet, and then can record single neurons. You can also use other types of electrodes, for instance, very similar to the one I showed you before, but now you have, let me see if I can show you the mouth. Yeah. You can have these little micro wires here popping out, and those can also record single neurons. And so to give you a concrete example of how this looked like, this is an experiment where the patient was listening to some words that correspond to objects, then had to decide whether those objects were bigger or smaller than a foot in size, because this was done in the US, and then had to click if it was true. And so what I'm going to show you is the words, the sound waveform that the patient was hearing, and the actual neural recordings. So the click you will hear is when this neuron, this particular neuron, is actually activated to that sound.
Elf. Frog. Oyster. Shelf.
So the little line going down that you see on the screen is where this neuron is actually sending action potential. And you can see that for some of the words it's not really responding,
Metal.
but some of the words actually responding more, sometimes very much, and sometimes really not doing anything else.
Cougar. Flag. Oyster. Shelf.
So what we want to understand is how do we make sense of this, right? We have this neural activating in kind of random fashion. For some words, we don't know why. What are the principles that we can use to try to understand this brain activity? And so that's the first part of my talk, where we look at different representations. What are these neurons doing to represent the different elements of language? So if we look at these recordings, and now I'm going to show you the same type of recordings for the word fork, but across many neurons. So this is for the same neuron, but across many repetitions of the same word, so many trials, and each of the dots correspond to an action potential. in time, and you can see this neuron is responding pretty well. But then if you look at all of the other neurons, you can see that some of them are not reacting, some of them are reacting a little bit, but we're not sure what they're doing exactly. So how can we make sense of this? And this is really the question that we want to figure out. And what we do is we try to not take a single neuron kind of approach, but really the approach of the population activity, right? To understand how all of these neurons together, they represent this information. And for this, to give you more details about how we do this, I give you an example with 3 neurons that we're recording for that patient. So the first thing we can do is to make it more simple, we can just smooth the firing rate so that we just get continuous lines. And then we build what we call a neural state space, where each of the axes in that space correspond to the activity of one neuron. So for these 3 neurons, we have 3 axes. And then if we take a single time window here, we'll have the firing activity for each of those neurons. So we'll get a single fixed point, so it's a single point in that state space. And if we move the window, then the point will move a little bit. And then if we move it even further and so on over time, we'll get a trajectory through that space. Of course, we don't have just 3 neurons, we have many more. So this is a very high dimensional space in which we have this trajectory. So as we go through time, potentially this trajectory could go everywhere in the state space. But what actually happens when you observe this trajectory is that they stay in a specific part of the space, and this is because the neurons are correlated with each other. And because of that, the activity will be confined to subspace, which is what I'm showing you here, that you can retrieve with dimensional interreduction method. And this is what we call a neural manifold. And what we are interested to figure out is how in this neural manifold, the trajectory are representing different elements of language, for instance, the phonemes, how to make sense of this. So this is the work that we did with Paavo Repic, who was a former postdoc in my lab. And using the same kind of data, we asked this question, how the different linguistic features were represented in those trajectories. So we first fit what we call a non-coding model, where we have all of these different linguistic features. And then for each of these features, so I'm showing you here, for example, with the phoneme K, we have the time, so at 0 is when the phoneme K is heard, and then each of the line corresponds to the activation of one different neuron. So we have 23 neurons here, and they all respond with different activation when it's yellow to that particular phoneme K. So of course, it's still a 23-dimensional state space, so we want to reduce the dimensionality. Here, for instance, using principal component analysis, and we get to 2 dimensions. And in these 2 dimensions, we now have a nice trajectory. So here you can see the numbers correspond to the time, so minus 100 milliseconds, 0 milliseconds, and so on. And what we want to understand is if this trajectory makes sense in terms of representing the phonemes. And so for that, we can use what we know from linguistics, right? So for instance, what we know for vowels is that there are different categories of these vowels. One way to classify them is to use the first formant, which is basically the first resonant frequency of the vocal tract, and which basically you can understand as the position of the tongue in the mouth. So if the position is very high, like for I or A, then it will be, let's say, for high first formant, the first category. And when the position of the tongue is lower, like A or R, then will be the other category. So I put them in red and blue. And if I take my trajectories in the neural state space for all of my phonemes, and I look how they evolve in time, so in blue is corresponding to the second category, in red for the first category, you can see there is maybe some kind of separation in that state space. But we actually want to quantify this, and to quantify this, we use something called a clustering index, basically just saying how well are those groups of trajectory separate from each other and well consistent with each other. And this is what we find in the end, so here in blue is a line of the actual data. The shading corresponds to the random or null distribution, if you want. And whenever this trajectory is red, it means that there is a significant encoding through these 2 different groups of trajectories. You can replicate this across many other features, like, for instance, the vowel second formant, or the consonant, the manner of articulation for consonants, of the place of articulation. And you can see that for some of them, for instance, for the manner of articulation, you find a very nice encoding of the clustering index. We also replicated this in the second patient, you can see here in green, and we can also find that those trajectories can encode as well, are meaningfully encoding those phonetic features. Okay, we then reproduce this across different paradigms. So here, what I showed you so far was for this task where the patient has to really take this semantic decision. But we also look in natural speech for those participants, and we also find that so some of these linguistic features, there was a nice, significant separation of those trajectories, and we also reproduce that in other patients. Okay, so why do we actually need single neurons, right? Maybe we could just look at the local field potentials that I told you and just try to make those analyses from there. This is actually what we try to do in this analysis by looking at the electrocardiography. So these are the sample you can see here on the brain, which record the local field potential while the uteri recording the single neurons was here. And what you find when you try to fit kernels of the phonemes, when you fit those models for the phonemes, that there is actually not much encoding, and in that case, nothing is significant. We also look at the phonetic categories, like the foreman, for instance, and we find that, okay, there are a few electrodes which show significant activities, but they are located very much closer to the early sensory processing, closer to the auditory cortex, if you will. But if the region where we're looking at, which is somewhat a higher level region, the activity is much sparser, and it's almost no surprise that we don't find it in the local field potential, which needs to assemble activity from many neurons. Okay, that was everything I had to tell you about representation. But as I said, now we want to try to understand how we can compose this representation and go from one level to the next, for instance, from phonemes to words and so on. And for that, I would like to introduce now dynamical systems and how we understand dynamics. We try to fit dynamics to the data. So if we just take a simple differential equation, dx over dt equal f of x, and x just represents in that neural state space, I was showing you the state of the system, right, where it is in that space. And we know that we want to figure out what are the conditions of evolution of this. So one way to understand this is really to look at the landscape where the state of the system is this dot that you can see here. Oops, sorry, here. And then the flow is going to be characterized by this function f and tells you where you're going to go. And basically what you will do is to go down this landscape, right? So if you're on a hill, you're going to go down the hill, and there is a valley here, you're going to go down that valley. So that's how we want to try to understand how composition is performed in the brain. And this is the work of my also former PhD student, Nusrat Mohammadi. And the first problem we have is that we are recording spiking data, right? And the spiking data are stochastic point processes. So it's not the kind of representation which are continuous differential equations that we can study with this landscape. So the first thing we need to do is to fit those models and to try to transform them into continuous differential equation. So we first fit statistical models, which are called point process models. And then what we developed is a diffusion approximation that allows us to change this stochastic point process into something much more continuous. And because of that, then we can have a set of differential equations that we can study with exactly the kind of tools I was showing you, the landscapes, if you want. Okay, so now we can feed those models to data and try to recover how this brain data are doing composition, right? So we actually do that for a patient, and the kind of composition I want to show you today is how we compose a single phoneme into bigrams, right? So two different letters into a pair of letters, right? With syllable, if you want. So this is an example for the word canoe. So we'll have a model fitting each of those different phonemes, but also the bigrams ka, un, and u, okay? So this is the model that we fit. You can see on the x-axis sort of the phonemes and the bigrams, and on the y-axis sort of the neurons, and this is the encoding weights. And also we couple those different neurons together. Now we can interrogate the model that we feed to the data and try to figure out what is the landscape and how this composition from single phoneme to bigram actually occurring. And before I show you the results, I would like to tell you three hypotheses. So the key idea is that, let's say you hear a first phoneme, you want to keep it in memory for some time, and until you hear the second phoneme, because if you want to compose these two things together, you need to remember what's the first one, so when you hear the second one, right? And this constrains the kind of dynamical solution we can find. So the first solution is to use what we call criticality during input, which means that when you hear the first phoneme, you will have some kind of flat landscape around that phoneme, and which means that the activity there will be very slow. It will be so slow that this can be retained in memory until you hear the second phoneme to compose the background. Another possibility is to say, I'm going to have something at criticality at rest, which means that when you return from the input, so after you release the first phoneme, you'll have a flat landscape again, and this can allow that can allow you to remember what was the first phoneme that you heard. And finally, a last solution is to say, well, I just have many fixed points, and I will just go to one or the other to represent the different phonemes and the backgrounds. So each of those solutions actually work for doing this kind of composition, and we then look at the model to try to figure out how the brain is actually doing it. So what we do find is that there is actually a single phoneme, and this is an example of the state space here that we reduce to 3 dimensions using dimensional reduction. And each of the dots correspond to the representation, the location of the single fixed point corresponding to one of the single phoneme, or when you use the background as an input. Then we want to find whether there is criticality, right, during input or at rest. And for that, we look at the eigenvalues of that fixed point. That's a traditional, classical way to do it. So this is, for instance, the eigenvalue for the phoneme T, the fixed point T if you want, and the eigenvalue for the background N. A sign of criticality would be that the real part of this eigenvalue, what you see on the x-axis, would be very close to 0. And this is not so much a case. Yeah, actually, you can see it's pretty far from 0. And if we quantify this systematically, so this is the real part of this, the maximum eigenvalue for this eigenspectrum, and the eigenvalue for all of the phonemes in the backgrounds, and you can see that it's never getting very close to 0. And it's also not going too close to 0 for the baseline. So basically, all my 2 hypotheses that I showed you before are also wrong because we don't have any criticality. It's what we call a hyperbolic fixed point, where there is nothing special about this fixed point. It's a bit annoying because we didn't really understand how is that possible that we can encode this background. But then we actually try to look more into details of the trajectories of how this is done. And so we looked at how the system is going towards the position of the fixed point during the input and how fast it's coming back. And so this is what I'm showing you is the rates of how fast you go and how fast you come back. Then we compare those rates between, how fast you go and how fast you come back. And so if you actually compare those rates, you find that for the single phoneme, so these are examples of single phoneme here, you will go there faster, slower that we will come back. But when you start encoding a background here, then you will actually come back slower that you will go there. And there is actually a dynamical scenario that can represent this, which is what we call a ghost attractor. So instead of having just a single fixed point, which is what we found before, so we still have a single fixed point, but the flat area is not really at the fixed point, it's near the fixed point, right? And this is what you see here. So basically the return dynamic will be slow, but will be, in the surrounding, in the area of the final fixed point that we have. And this scenario is actually a bit like the middle scenario that I was showing at the beginning, but it's actually more resilient, more stable to noise. And so this is much more generic way for the brain for doing it. So we thought it was a very good, very interesting finding. Okay, we're trying to extend this notion of composition beyond the cortex. So this is an example that we really recently finished the work on of an experiment where patients are saying different strings of letters, and we record from the deep cortical structures now, like the entorhinal cortex and so on. And what we do find is that we compare the encoding for single letters and pairs of letters, so 2 letters together. And we do find that some neurons are actually responsible for encoding those pairs of letters. And then eventually the idea would be to go towards population dynamics as well. So far I told you about composing phonemes into syllables or backgrounds, but we want to go toward more complex types of compositions, in particular syntax. And this is here an example of an experiment we started to do in the lab. So here you can see an image that we show to the patient, and the patient has to say what he sees on the image, right? So he would say, I see trees and fish with crosses. Then we show a certain kind of image, and for this image, the patient will say, I see trees and fish with crosses. So these 2 sentences are exactly the same, which is the point, because then there is no confound. But what's going on in the brain of the patient is different, right? Because in one case, the cross, if you want to have parentheses around the fish and the cross, they are grouped together. While in the other case, you first group together the trees and the fish, and then you add the cross on top of it. So there's a different operation there going on. And so what we do is we record from different patients, and here I'm showing the results for electrodes in the temporal lobe. So you can see here over the time the different words that the patient is saying, trees and fish with crosses. And then the blue condition corresponds to that image and the red condition to that image. And you can see at the beginning there is no difference. But as we go towards fish with crosses, then you can see difference between those time series. And so we are trying to better quantify how this is actually occurring and what's going on. And to try to understand that better, the idea is to not just stay at this high level of the just describing the composition of the syntax level, but to go a bit more formal towards what are the primitives, what are the operations that allows us to do this kind of compositions. And so for that, what we do is we look into this neural state space and we say, let's say we have a representation for two elements, vector A and vector B. And what we want to figure out is what are the compositional operations that allow us to actually do this composition. And this example, for instance, A plus B or A times B, this will be two primitives that will allow to do this. And what we try to understand now in my lab is to really see how this would evolve through time, so through the neural dynamics, and to understand how these different trajectories or these different operations actually map out on this neural manifold in the subspace. And this is here an example showing you how you would compose cat with different piece of trajectory for each of the phonemes. Okay, so I'm done with the composition part, and now I will talk a little bit about the interactions. And so I'm coming back to the work of Pavo here. And what I didn't tell you so far is that we look at the representation of single phonemes. We also look at the representation of semantic. And this is an example of a natural conversation we had for these two participants. And we could also, in that case, look at the different semantic contents of the word that were held by the patient. And this is an example for these two patients. Doing the same process that I showed you before, trying to feed those models, reduce the dimensionality, and looking at those trajectories, we find that depending on the semantic content now, we find separation between those trajectories. And these are the red area you can see here and here for these two patients. Okay, so now we have basically in the same population of neurons, semantic content and then phonetic content. And these are two different spaces, right? It's kind of multiplex in a sense. And what we want to understand is how these things interact together, right? What are the connections between those representations? And basically for that, there are two possibilities. Either it's something bottom-up, so it's like the phonemes which are built together to make words, or it's something top-down where the semantic content is going to influence what are the phonetic representations we'll see. So for the bottom-up cumulative hypothesis, let's say you hear the sentence, when is your, and each of those words has when is your, and you can look at the different phonemes there. The semantic would happen always at the same time after the word onset. This is what I showed you before. So basically, after when, after some point, you will have the representation in the neural activity of the word when, and so on and so forth. But now for the phoneme, we want to cut that into different components, which depend on what is the order of the phoneme in the word. So first we look at the first phoneme, and let's say we hear the first phoneme for the word, W. When we hear this one, as I said before, we want to keep that in memory until we resolve the semantic, right? So basically we'll hear this phoneme, and then it will stay there for a long time until the semantic is resolved. And then for the second phoneme, which comes a little bit later, then we also want to keep that in memory until the semantic is resolved, and so on and so forth. So we'll really see this hypothesis would lead us to this kind of scheme here. Now there is another possibility, which is the top-down predictive approach, which is basically you start from the same semantic representations, but then now the phoneme, if you look at the first phoneme, instead of having this build-up, we think in that hypothesis that it would be driven by the semantic content, right? So the semantic would basically be built either from context or from another processing stream, and then would come back to this area and then say, okay, now I want to compare what's going on with what I think it is with the actual phonemes that you heard, and therefore replay those phonemes. And that's what happened for the first phoneme and also for the other ones. So we get this nice alignment of all of those phonemes independent on their order. And so we check that in data. So these are the same kind of patient data, and this is for the first participant. So this semantic kernel is what I showed you before. And now if you look at the first phoneme, you can see that there is this kind of late activation. We can then look at the second phoneme, and we see also this very nice late activation, kind of at the same time. And looking at all of the phonemes, we find this very nice, neat alignment that would correspond to the top-down hypothesis. We actually, which is there. We actually check that in the second participant, and we also find this very nice and neat alignment across all of these phoneme order. So this would very much validate the top-down hypothesis and top-down prediction, at least in that particular brain area. We are not trying to extend these results across different brain regions to look also at other level of the language hierarchy. And for that, what we do is we record from different brain regions, and we try here, in the context of phoneme, to decode the different phonetic group, right? So this is an example of this. Over time, we say we want to decode, let's say, between these two groups of formants I told you about, and we look at how long those decoding values are significant. And what we want to figure out is, maybe depending on the brain area and the function of that brain area, how long and when we can decode from these brain regions will change. Because if your phoneme is used, I don't know, just for very quick recognition, it will stay for a very short time. But if it's used for building a very large word, then it would need to stay for a long time, right? And so this is kind of what we find. We find across those brain areas, example here for one patient, that the onset, of course, happens at different time, but also the duration, how long these phonemes are encoded, is going to change as well. Of course, when we look at these different brain areas, they need to communicate together. And that means that this local field potential, they need to propagate in some ways. So we can look at now this grid and looking how the local field potential propagates throughout these brains, but also at the uterine level. And you can see a little movie where you can see this propagation of the brain activity or the local field potential throughout the array, and basically showing how this information could be transmitted throughout those different areas. And we actually find that this is relevant for language because the phase of those patterns actually correlate with the spiking activity of those neurons. So let me return to where I started today, which was the sentence, a retired neuron politely declined the linguist who had organized a fun evening. And I try to show you today a little bit how can your brain understand all of this sentence under a second. And basically the different three levels I showed you, which is the representation that linguistic features leave as trajectories on these neural manifolds, that we can compose these different features and particularly try to understand how this is done from the perspective of dynamical systems on the neural manifolds, and then how these different features interact and this cross-level alignment in real time. And so the kind of model I showed you is part of a larger set of models that we can use in language neuroscience and that we detailed in a recent review paper, where basically we can think about those models along two axes. The first one is the covariance dependency, and the second one is the temporal dependency. And so the most simple models that people use in language neuroscience is the are called the multivariable static models, where basically you just take one feature and you relate it to one particular neural feature, and you can really well identify how these two relate at a given time point. Then you can make it more complex and try to look how these different features, either neural or auditory, interact together as a network to represent this information. You can also look at the temporal dimension and say, how is my feature actually evolving through times? And the kind of model I showed you today is really the last quadrant here, which is really those kind of dynamical multivariate models, where basically we have some kind of complex patterns, correlation patterns across the neurons that evolve through times in a latent space. And this is really the way we are interested in characterizing those dynamics and representations. There is one other axis of research that we're interested to tackle in the lab, which is basically learning, in particular learning to compose. And so if you take an embedding space, it could be neural activity, it could be something else, and we look at this space. If you look at these two concepts, dog and cat, in the sentence, the cat is chasing the dog, basically you don't learn, you have to learn through your lifetime how to put some particular rules to these different elements. For instance, you have to learn that the cat is a subject, an agent, and that the dog is an object or a patient, right? It's not innate. And maybe those rules actually are using some kind of operational primitives, as I showed you before. And so we are interested to understand how this can emerge through different neural networks and in the brain. And so we'll actually simulate those, and then we'll compare those to the actual neural data. Okay, so just to conclude and wrap up, the question that really drives the lab is how does the brain compose meaning from sound? And we look at different levels of language that I showed you today, the representation, composition, and interaction. And eventually the goal is to try to compose the different things together into a single model that can really help us understanding the processing of language in the brain. And so this is what we do in the lab. We try to understand this from the perspective of neural population dynamics, and what we study in the neural dynamics lab. And I want to mention here that it's not my work, it's the work of all of the people in the lab. And so my first thing, I would like to address them to all of the people in my lab, which are all very talented people and really makes this possible. And in particular, I would like to highlight the work of Pavo Orepic and Nosrat Mohammadi, who are the first two people in the lab here, and I feature a lot of their work today, but also all of the people now working on this compositionality and learning. So I really would like to thank you for all of your hard work. I would like also to thank my mentors, in particular Victor Iosa, who was my PhD supervisor and really taught me how to see the world through the lens of dynamical systems. Wilson Trucolo, who taught me how to fit those systems to the actual data through statistics. And Ligeo, who taught me how to actually ask the right question. And Nina Kazanina, who taught me what linguistics really is. So. And I would like also to thank the patients. These patients, they have epilepsy, they come to the hospital, usually they're in pain, it's a difficult time for them. And yet most of them, they accept to participate in those experiments. And even though they know that there is no benefits for themselves, but just knowing that this could help research, fundamental research, and potentially some patients in the future, they actually accept to participate. So there would be no data without them, so I would like really to thank them, as well as the clinical teams that help us to record those data, and all of my collaborators. So here I put only the PIs, but of course there are all of the students and the members of those labs that do the actual work, so I would like to thank them as well. And finally, I would like to thank the Institute of Bioinformatics, INI, which for hosting me. I think there are a lot of very wonderful people there, and really it's made by the people what INI is, and it's really an honor to work with you. I would like to address a special thanks to Ruth for organizing all of the apéro here, so it's thanks to her that you have an apéro today. And I would like to thank ETH Zurich and the institution that supported me throughout my career, the founding agencies, because there will be no research without them, and my family, my parents, and my friends for supporting me, and you for your attention.